We explore how genetic studies reveal the Arabian, nomadic, and colonial influences in Somali's DNA. We uncover surprising insights into the history, ancestry, and resilience of the Somali people, offering a unique perspective on human migration and cultural exchange. Further, whole genome analyses show that Somalis share more genetic affinity with Southern Arabians than with Northern Middle Eastern populations. This narrows the likely origin of these ancestral contributions to Yemen and Oman, coastal regions with long-standing maritime traditions. It is probable that many of these ancient contacts were tied to the seasonal monsoon wind system, which governed when and how boats could cross the Red Sea. The Arabian traces in Somali's DNA are also preserved in the linguistic and cultural domains. For instance, Arabic words have entered Cushitic languages over centuries, especially in religious, legal, and commercial contexts. Oral traditions, poetry, and even clothing styles in Somalia bear influence from Yemeni and Omani customs. Genetics merely affirms what history and anthropology have long suggested, that the sea was not a boundary, but a bridge. Modern Somalia continues to reflect this hybrid identity. Genetic studies show that the Arabian component in Somalis is older and more deeply integrated than many realize. Unlike recent migrations, which often form distinct genetic layers, these Arabian contributions are evenly distributed across families and regions, evidence of integration rather than colonization. Importantly, this genetic blend does not diminish African heritage. Instead, it enriches it. Somali's genome is a record of exchange, continuity, and complexity. Its Arabian strands are part of a larger story of how humans crossed water, mingled genes, and created societies rooted in shared experience. The story of Arabian ancestry in Somali DNA is not a tale of conquest, but of cooperation. It tells of dhow sailors crossing currents, of farmers seeking greener pastures, of marriages that joined lineages across sea and sand. It is a story etched in the spiral of chromosomes, one that reaches across generations to connect Africa with its neighbors. Somali's DNA, then, does not draw sharp lines between continents. It blurs them. And in that blurring, we discover not confusion, but clarity. A deeper understanding of what it means to be human in a world where land ends. But history does not. Across Somalia, Djibouti's arid plains and rocky highlands, the nomadic spirit has long defined survival and identity. For thousands of years, pastoralist communities have crisscrossed this rugged landscape, herding goats, camels, and cattle in search of water and grazing land. Yet beyond their way of life, these movements have left a deeper, less visible trail, one encoded in the very DNA of their descendants. Nomadism, often misunderstood as random wandering, follows established seasonal rhythms that reflect generations of environmental knowledge. In Somalia, these seasonal migrations created patterns of interaction among clans, ethnic groups, and even outsiders. Every movement meant not just the exchange of goods, but the mingling of bloodlines. Over centuries, this formed the unique nomadic genetic network, connecting people across distances where no cities existed. Genetic sampling among nomadic Afar and Isa communities reveals patterns of gene flow that don't match rigid geographical boundaries. Instead, their DNA tells a story of constant exchange across East Africa, Western Ethiopia, and Somalia. These connections span desert corridors, highland passes, and dry riverbeds, routes once traveled by barefoot herders and caravans laden with salt and incense. One of the most telling examples of nomadic gene flow is found in the relative uniformity of certain Y chromosome markers across wide areas. Haplogroup E1b1b, especially the subclade EV32, appears prominently among the pastoralist populations of the Horn of Africa. This lineage likely spread through male-mediated migration among herding societies. Its widespread presence underscores the social organization of nomads, where male inheritance and patrilineal clan structures guided the transmission of land, livestock, and legacy. Interestingly, autosomal DNA, representing a mixture from both maternal and paternal ancestors, paints a more varied picture. It shows that while patrilineal identity might remain constant, maternal genetic contributions were more diverse. This suggests that marriages often took place with women from different clans or even different ethnic groups. It was a strategy that brought new alliances and adapted communities to ever-changing environmental realities. Such practices created what geneticists call a colonial gradient, a gradual change in genetic frequencies across a landscape rather than sharp breaks. In Somalia, this gradient is particularly visible in regions where nomadic paths intersect with sedentary populations. The more isolated highland communities tend to preserve older genetic signatures, while those along traditional caravan routes display more genetic diversity. These dynamics also influenced health-related genes. For instance, certain gene variants associated with resistance to dehydration and efficient fat metabolism appear more commonly in nomadic groups. These adaptations, selected over generations in extreme heat and scarce water conditions, help shape the genetic resilience of these communities. Even today, metabolic studies of Somalis and Djibouti and nomads reveal unique physiological profiles optimized for energy conservation and endurance. Lactase persistence, the ability to digest milk into adulthood, is another genetic trait closely linked to pastoralist ancestry. In Somalia, Djibouti, this trait is found at a higher frequency among nomadic populations than among coastal or urban groups. This makes sense in a society where camel and goat milk are not luxuries, but staples of life. The presence of lactase persistence genes provides a living marker of dietary adaptation and cultural continuity. But the genetic legacy of nomadism is not only biological, it's historical. Oral traditions among clans often trace lineage back hundreds of years, and sometimes across borders. These stories, once dismissed as folklore, now find echoes in genetic data. Researchers mapping family trees have discovered a surprising correlation between oral genealogies and actual genetic lineages, especially on the male side. This validates the historical memory encoded in clan structures and supports the idea that Somali nomads have not been aimless drifters, but archivists of movement, holding maps in their minds and legacies in their blood. However, the pressures of modern borders and sedentary policies are altering these ancient patterns. Nomadic lifestyles are increasingly constrained by national boundaries, environmental degradation, and conflict. As herders settle or are forced into cities, the genetic diversity shaped by centuries of movement may begin to narrow. This raises urgent questions about what will be lost, not just culturally, but biologically as mobility fades. Yet the genetic record already holds much of this history. By sequencing the genomes of elderly nomadic individuals and preserving their stories, scientists can archive a way of life that helped define the Horn of Africa. Nomadic DNA carries echoes of drought and rain, of marriages forged over shared watering holes, of alliances sealed on sun-scorched plains. In recent projects, Djiboutian researchers are collaborating with mobile health teams to ethically collect genetic data from remote nomadic groups. This ensures that their knowledge, traditions, and biological heritage are not only respected, but included in broader narratives of human history. The focus is not extraction, but preservation, a reciprocal effort to keep the memory of movement alive. Ultimately, the story of Somali and Djibouti's nomadic DNA is a story of adaptation in motion. It reveals how humans have not merely survived harsh environments, but thrived through movement, flexibility, and connection. These are lessons embedded in the genome, reminders that stability is not always found in stillness, but sometimes in the steady rhythm of footsteps over sand. As science listens more closely to these genetic whispers, we begin to understand that nomads were never on the fringes of history, they were its carriers. and in the rolling dunes of the Horn of Africa, their genes still walk with them. When the French colonial forces arrived in the territory now known as Djibouti, the late 19th century, they came with maps, treaties, and an imperial vision. But they also brought with them more subtle legacies, ones that would eventually be written into the DNA of the people. Unlike the more overt political and cultural impositions of colonialism, these genetic imprints often went unnoticed. Yet their effects linger in bloodlines, health patterns, and even ancestral identity. Djibouti officially became French Somaliland in 1896, and for nearly a century, the territory served as a crucial outpost for French imperial ambitions in the Red Sea. With the construction of a railway from Djibouti city to Addis Ababa, thousands of laborers, administrators, soldiers, and traders moved into the region. While the French presence was initially limited to military and commercial interests, it gradually expanded into local governance, education, and settlement. This influx of outsiders, primarily French Europeans, but also colonial troops from North and West Africa, introduced new genetic material into the population. Genetic studies of modern Djiboutians, particularly those from urban centers like Djibouti City and Tajura, show low but detectable levels of European and West African ancestry. These aren't dominant in the gene pool, but their consistent appearance in certain autosomal markers suggests admixture events that coincided with the colonial period. Interestingly, Y chromosome data reveals more about the male-mediated aspects of this admixture. Lineages commonly found in southern Europe, such as R1b, appear in trace frequencies among some Djiboutian men. This hints at French male settlers or military personnel who fathered children during their time in the colony. Yet the limited spread of these markers indicates that while such unions did occur, they were neither widespread nor sustained across generations. On the maternal side, mitochondrial DNA shows even subtler signals. Haplogroups associated with West African lineages, particularly L2, are present in Djiboutian urban populations at levels higher than expected from regional migration alone. This suggests the presence of West African women in the colonial era, labor or domestic service networks whose descendants became absorbed into the local gene pool. These findings reflect broader historical dynamics. Colonial administrations often employed soldiers from other colonies, and Djibouti, being a strategic military post, saw the arrival of troops from Senegal, Mali, and Madagascar.